Welcome everyone. Today we are going to learn about transcellular transport of glucose. Now, why it is called transcellular? At first, I want to mention that this cells is actually our brush border cells that lines the small intestine. So it is present in the small intestine. Now, this cells actually helps in the absorption of nutrients into the cell from the small intestine. So these portions, the outer portion of this cells is called lumen. Now, these cells has polarity. These portions where the microvilli are present are called apical portions, apical portion of the cell. And the bottom portion is called basolateral position of the cell. The apical portion of the cell faces the lumen where nutrition is present, and the basolateral domain of the cells actually face the blood. So here blood is present. Now, in the apical portions of the cells, there is a particular symporter present that is called sodium glucose symporter. Sodium glucose symporter. I have made a separate video on what is sodium glucose symporter and how it works. So I will not actually describe here the details of this symporter. But what happens? Sodium actually two sodium ions enter through this symporter inside the cells. Why? Because the outside concentration of sodium is high and the sodium concentration inside is low in the cell. Okay. And glucose here actually moves from lower to higher concentration. The outside glucose concentration is low and inside glucose concentration is high. For these cells, glucose concentration in the inside is high. Now, the sodium concentration actually keeps low by the presence of ATP driven pump called sodium potassium pump. Suppose this is the sodium potassium pump. By working this pump, what happens? Sodium ions, 3 sodium ions are actually export out of the cell and 2 potassium ions import occur. As you can see, sodium ions are moving out of the cell, so the concentration of sodium remains low, and that's why sodium actually can enter inside the cell because of the presence of dietary sodium chloride, the salt. So sodium actually comes out. Now potassium actually, this potassium, when it moves inside the cell, so the concentration of potassium increases. The concentration of potassium increases. And in this basolateral position, there is a potassium channel present called leaky potassium channel. Through this, potassium actually moves out of the cells, and by the working of these 2 sodium potassium pump and chlorine potassium channels, there is the establishment of that is called membrane potential or resting membrane potentials. Resting membrane potential. Now, what is the role of resting membrane potential? As I have mentioned, that sodium potassium pump actually established that there is a low concentration of sodium, so sodium can enter from higher to lower concentration, and this moving of the higher to lower concentration of sodium gives the energy for the movement of glucose from lower to higher concentration. Because in my previous all videos I have mentioned that there is 2 types of movement occurs in for the molecules. One is called, suppose this is the hill. So what we know that when someone comes down of the hill, it is called downhill movement, and during downhill movement energy is not required, but during climbing up of the hill it requires energy. So this is called uphill movement. Molecules when move from higher to lower concentration, this is called downhill movement. Sodium showing the downhill movement, but molecules when move from lower to higher concentration, it is called uphill movement, and here energy is required. Like the glucose concentration outside is low and inside is high, The movement of glucose is called the uphill movement, and this requires energy. Sodium movement gives the energy. In my sodium glucose importer, I have actually mathematically shown how much energy is actually generated when sodium actually moves through this importer, and through this energy glucose import occurs. Okay, now the sodium potassium pumps generate the sodium concentration gradient, that is inside is low, outside is high. But with the help of potassium channels, there is a resting membrane potential, and during this resting membrane potential, what happens? The outside of the cells remains positively charged, more positively charged than inside. So inside is negatively charged. That's why more sodiums are actually attracted, because sodiums are positively charged and they are attracted towards the negatively charged. Okay. Not only that, there is a unipolar in the basolateral position that is called GLUT2 proteins. Through these GLUT2 proteins, this glucose actually moves out of the cell into the blood. As because glucose are moving from apical portion to the basolateral portions into the blood, that's why it's called transcellular transport of glucose. So glucose actually coming from the nutrition to the cell, and then it goes out of the cells through the GLUT2 proteins. This is an unipolar through which glucose actually moves into the blood. Now through this diagram, I can also explain how one important thing that is called oral rehydration solution, actually works. Now, why oral rehydration solution is given and what is oral rehydration solutions? As because its name implies that rehydration. So when dehydration occurs of our body, then this solution is given through the oral or orally. Now, when dehydration occurs during the infections, bacterial infection in the small intestine, like cholera bacteria infections, then dehydration can occur. What happens when cholera bacteria infects these brush border cells? These cells actually secrete huge amount of chlorine outside the cells. So chlorines are secreted out through the chlorine channels, and huge amount of chlorines when move into the lumen of the small intestine, then water actually flows from blood to the outside or lumen of the small intestine through the tight junctions. These all epithelial cells are connected with tight junctions, and water moves through the tight junctions into the small intestine and excreted out. Now, just by taking water, it will not solve this problem, because as soon as water will be taken from this lumen, water will be excreted out. So scientists actually developed this oral rehydration solution by observing this phenomena that if sodium concentration is high, if glucose concentration is high inside these cells, then one thing occurs that is called the increase in osmotic gradient. This is called transepithelial increase in osmotic gradient. So when sodium, chlorine, potassium, glucose, these molecules and ions are inside is higher concentration, then water actually flow through this tight junction into the blood. So in the oral rehydration solution, sodium chloride, potassium chloride, glucose, these are actually mixed and it dissolves into the water. So water here it comes, and through the channels like sodium, chlorine, I have mentioned that through the importer, sodium, glucose actually enters, and by this way the osmotic concentration or osmotic pressure gradients increases inside the cells, and water then actually, as because the osmotic concentration gradient or osmotic gradients increases inside the cells, so water actually moves through this tight junction into the blood, and that actually prevents dehydrations, and also it can cause diarrhea and ultimately death. So one thing also has to be done, that is the treatment with antibiotic, so that the bacteria can be killed, and as well as oral rehydration solution should be given to the patient. So how this solution works? By the presence of this importer and presence of sodium potassium pump, chlorine pumps and GLUT2 proteins. through all this orchestration of all the transporter, glucose, sodiums are actually moves in, and that's why scientists observe this phenomena. So they have chosen sodium, chlorine, potassium, and also with glucose that actually absorb into the cells, and that increases the osmotic gradient, and water actually flows through these tight junctions into the blood that prevents dehydrations and cause rehydration.